Hello everyone, my name is Khushi and I am a member of Shivi Forensic. Today we are going to discuss about what is capillary electrophoresis, its principles, its components, working, advantages, disadvantages and forensic significance. Okay, the first that we are going to discuss is what is electrophoresis. Electrophoresis is a physical method of analysis. Okay, or we can say electrophoresis is a laboratory technique that we use to separate charged molecules like DNA, RNA, or protein based on their size and charge by applying an electric field through a medium such as gel. In this diagram, we can see some forces are acting on a particle. So, these forces are electrostatic force, frictional force and net force. Okay, three forces acts on a charged particle, that are electrostatic force, frictional force and net force. So electrostatic force pushes charged particle or molecule through the gel. Okay, frictional force opposes the motion as molecule move through the gel matrix, and net force allows molecules accelerate until the electrostatic force equals the frictional force, that results in the constant velocity. Okay, so next is capillary electrophoresis, and that is our main topic. So basically capillary electrophoresis is an analytical separation technique based on the movement of charged particles in an electric field through a narrow capillary tube. Okay, earlier we used to use gel electrophoresis, as gel electrophoresis is a traditional technique, but it is very slower and not efficient. But now we have capillary electrophoresis that is much faster and more efficient than gel electrophoresis. Why it is more efficient? Because it uses thin capillaries, okay, less than 100 micrometer in diameter, and that allows better heat dissipation and sharper separation. So basically this is the diagram of capillary electrophoresis, or we can say working of capillary electrophoresis. So on the left side we can see cathode and source reservoir, on the right side we can see anode and destination reservoir. So basically capillary connects the source reservoir to the destination reservoir. In the mid of this there is a detector that detects the signal coming, and all this is connected to the power supply. Okay, so now we will discuss about principle. So basically when a thin capillary tube is filled with buffer solution, then the sample is injected at one end and high voltage is applied. So the charged molecules will move according to the speed and depending on their charge. So basically some molecules will move towards the positive end, that is anode, and some will move to the negative end, that is cathode. Second point is according to size. So the molecules that are small in size will move more swiftly, means it will move more faster than the larger size one molecules, because smaller molecules can travel through the pores easily than the larger molecules. So in short we can say that capillary electrophoresis separates molecule based on how fast they move in an electric field inside a capillary. Next are components that are used in this technique. So first is capillary tube, that is usually made of fused silica with internal diameter of 10 to 100 millimeter and 20 to 100 centimeter length. Second is buffer or electrolyte solution that fills the capillary and reservoir. It maintains pH and ionic strength, controls osmotic or electroosmotic flow, and ensures reproducible separation. Next is high voltage power supply that provides a strong electric field for separation. Next is sample introduction system or sample injection by pressure or vacuum, or electrokinetic injection that is used to introduce a very small volume of sample into the capillary. So next is detector, or we can say detection system. So most commonly used detector are UV visible absorbance detectors. As we know, fused silica capillaries are transparent at less than 200 nanometer. So for visibility we used UV visible absorbance detector. And alternatively we can use fluorescence, conductivity, mass spectrometry, electrochemical detection. Next is data acquisition system or computer, in which all the data is visible and recorded. So the next is the working of capillary electrophoresis. First step is capillary preparation. In this, capillary is first filled with a buffer solution like sodium dihydrogen phosphate or NaH2PO4 that act as a medium for separation. Then we take two electrodes and place them in the buffer solution at both ends. Second step is sample injection. In this we take tiny amount of sample and introduce it in the capillary inlet, that is near the cathode. This we can done by two processes. First is electrokinetic injection and second is hydrodynamic injection. In electrokinetic injection we apply voltage, and in hydrodynamic we apply, or we do it by using pressure. Third is voltage application. We apply a high amount of voltage across the capillary. We generate two forces. Electrophoretic force, what it does, it moves analytes based on charge and size. Okay, a small size particle will move faster, and large size particle will move slower. Second is electroosmotic flow. It moves all components towards the detector. So our fourth step is analyte separation. As we know, each component migrate at different speed. On what it depends? It depends on charge and on the size of the particle, and also on the direction of EOF, that means electroosmotic flow. And as they move, they get separated within the capillaries. As a smaller particle will get separated easily, larger particle will take some time. Fifth step is detection. As the separate analytes exit the capillaries, they pass through the detector. Usually we use UV visible detector or fluorescence or conductivity, and then each compound will produce a signal. That signal is visible at the computer. Okay, how they will be visible? That signal will be converted into an electrophoregram, a plot that will show peak and time. Each peak represent different analyte. So next are the advantages. So first is it requires very less amount of sample. Second, it takes short time for the analysis of sample. Third is it is simple and automated technique. Fourth is high efficiency of separation takes place in this technique. Disadvantages are like simple molecules may get stick to the wall. Simple molecules are like water, small gases, and some certain ions also like sodium, potassium, calcium, chloride. They can get stick to the wall and do not get separated. Second is the result obtained are not much reproducible. So further we will discuss about forensic significance. So first is DNA profiling. So primary use of DNA profiling in forensic science is basically STR analysis for human identification. Second is drug and toxicology analysis. So basically capillary electrophoresis separates and identifies charged drug molecules. Example is cocaine, amphetamines, opioids, hallucinogens, etc. in biological samples. It is also used for seized drug analysis, like for purity testing, adulterant detection. Third is ink and dye analysis. In this capillary electrophoresis helps forensic scientists differentiate ink on question documents. Example like checks, contracts, wills. Like it can detect whether different inks were used on the same document. It is also used for separation of dyes, like inks contain mixture of dyes and pigments, so it is also used for separation of dyes. Next is protein and peptide analysis. Like capillary electrophoresis separates peptide based drugs like synthetic opioids, peptide hormones like growth hormones, or erythropoietin in doping cases. It is also useful in sports doping control and toxicology. And last is explosive residue analysis. This is all about capillary electrophoresis. Thank you.